This part of the presentation extends on what Matt just described from an introductory perspective. So the technology is the same, the methodology is the same. It's a difference in application. And that application difference is going from clonal sequencing in the case of protein engineering and now to Meta-Transcriptomics. And this was done in collaboration with colleagues at New England Biolabs, and we'll recognize those folks in a couple of minutes. A couple quick slides for introduction. This is just orienting Meta-Transcriptomics to metagenomics. And as we all know and have seen over the last several years, the use of NGS approaches in the deciphering of complex bacterial mixtures, whether it be for microbiome, environmental metagenomics, et cetera, that has become very routine and very, very common. In fact, before joining Element, in collaboration with Chris Mason, we've done a wide variety of urban metagenomics through the Metasub project, which has become a worldwide effort. So the point being is that next generation sequencing approaches have been very, very efficient at helping understand the complexity that's represented on the left-hand side, on your left-hand side of the slide, by an oral microbiome indication of a variety of different species. In the case of DNA or metagenomics, that DNA is isolated, library prepped, put on the sequencer, and then analyzed, where you have insights into the identity of who is there, right? What species are there, and the potential that those species have based on what we know about the species. In the case of Metatranscriptomics, it's a slight shift in that instead of analyzing DNA, we're now looking at RNA with the idea of identifying what activity is present. So essentially, not just the species that are there, but what relative activity of transcription they are undergoing, and giving an indication of what the activity of the actual species is there. And of course, which one of those is more effective, more approach, or perhaps both, is entirely dependent on experimental design. So what we'll focus on is a couple of challenges that exist, and then how the LoopSeq technology can be applied to those challenges to help address them. So generally speaking, with short read technologies, as we know, whether it's some length up to approximately paired N150, that was the comparator data set in this case. And I also want to make the point, the next several slides that I'm showing were from Lawrence Etwiller and colleagues at NEB. So this is not elements analysis, this is analysis from NEB, and showing some examples of, from a complex mixture of bacteria, and then isolating RNA, performing RNA-seq, and then mapping that onto, in this case, one example region of a bacterial genome, and then asking what is the mapping quality and what is the unique mapping ability. So what's shown in the brighter blue and red is the forward and reverse reads, and in the brighter darker color, those are mappings that are correct and that were correctly mapped to in an individual point in the genome. And then anything shown in the lighter color or very, very dim colors indicates mappings that had very, very low quality or were mapped in multiple locations, meaning that there was not enough information for the short read to be uniquely mapped to a single point in a single genome. So and this theme will continue through some of the other example screenshots from IGV, but just wanted to orient to what those colors represent. So the brighter color, correct individual mappings; the lighter color, it just indicates not that there's anything wrong with the sequence, and that's a key important point. It's that there wasn't enough information in that sequence to uniquely map it to a single point, meaning we needed more sequence information along that transcript to uniquely map it. So NEB developed a method called CappableSeq, which is shown on the left-hand side of the slide. Essentially, it takes advantage of the 5' cap of a transcript, as well as the poly A tail, with the idea that you can capture a full-length transcript and then sequence that transcript by long-read sequencing. So I won't go into the method in detail other than to say this is a way to capture full-length transcripts from bacterial species in this case. Shown it with some graphically on that left-hand side. A reference is given on the bottom and can go into more detail. And then if we compare the PacBio data, in this case, the CappableSeq was developed for use with smart sequencing. And some of the results are shown on the right-hand side of the slide. So you see some of the PacBio reads in the lower panel versus short reads in the upper panel, indicating the very clear performance of those longer reads. So it's probably no surprise to anyone, by capturing those very long reads across those long transcripts, you can gain enough information to now uniquely map those to a single point even in a complex mixture. So the question that we were asking, now that I've kind of completed this introduction, is can we take LoopSeq technology and use the CappableSeq method to perform the capture of the full-length transcript, but then rather extending that through to smart sequencing on PacBio, use the LoopSeq technology, and then use the short-read sequencer to take advantage of all of the cost and performance and throughput advantages that Matt just described. So those same themes exactly apply: lower cost per G, much higher throughput, et cetera. In this case, the question is can that be applied to the CappableSeq? So you saw this slide a moment ago. Same, again, emphasizing same technology, same methodology that's being used here. So I won't go through that. And here we'll have a summary of the results, again, provided by NEB. And I think there's a couple of key methods. So, kind of a prelude to the conclusion. You see LoopSeq kind of off by itself on the right-hand side. And this is showing both reads that are unmapped as well as reads that are mapped. And reads that are unmapped in yellow, and then reads that are mapped to a reference genome shown in green. So the ideal case is those should be relatively equal if the information content and the ability to map those is the same between the cases. And the other graphs going from left to right is nanopore, and then nanopore with correction, and then PacBio, a nanopore with Illumina plus another form of correction. So you can see with read correction, things get better as we go across. In the case of PacBio, it's self-corrected with Canoe, which is shown on the lower panel, and then in nanopore corrected with either Canoe or some other methods, which is shown on the upper panel. So it means that the long-read sequencing can certainly improve with correction, but importantly, the LoopSeq method is shown without any correction whatsoever, other than going through the process that Matt described, that individual de novo assembly that happens on the read clouds, and then that assembled molecule then being able to be mapped. So there's not a requirement for a reference sequence to perform that assembly ahead of time, and then you can see the significant increase in performance. Now, the ratio of ORF to read length, which is the axis on one side, that's a bit of a theoretical representation of this. We've been talking with Lawrence at NEB, and the reason why it's centered around that 0.75 and why it doesn't rise higher is again because of a theoretical versus observed difference in the process and the way that these plots are developed. So if we now look at how well does it capture abundance in mixtures. And so this experiment was done directly. So you see 3 or 4 different species, 3 of them E. coli, 1 a lactobacillus, and shown the relative ratios that were in the mixture as well as the growth medium that was used in each case. So LB for most, a rich media for 1. And again, the idea is how well does the transcriptional activity recapitulate the mixture? And there's an important consideration that, remember, if we were doing DNA, we would expect that to be a perfect mixture. If we're doing RNA, we would expect to see a representation of activity. So we're not expecting that measurement to necessarily be precisely representative in that order. But a couple observations were made. So the dotted line indicates an expected 1-to-1 correlation of gene abundance if we looked at an E. coli strain by itself, meaning it was done in isolation, and then asked, can we pull out those transcripts from the mixture alone? And you can see in this, there's a higher abundance in the mixture shown due to erroneous mapping. This plot was created from short read information, and that higher abundance in the mixture, again, is indicated by that erroneous mapping. So we're counting things to more than one species with the short read data. If we look at the LoopSeq, which is shown on the top, which in the mixed population versus the same short read only, you kind of see that same mapping problem we saw before. So a much cleaner representation. And then if we ask the transcript mapping of the strains and mixture, the expected value from the mixing ratio, again, is shown with the horizontal bars. And then whether we use Loop or Illumina, and you can see the relative difference in the transcription. The key point here is not necessarily that distance from one to the other, because remember, this is an activity measurement, not necessarily a DNA abundance measurement. But again, the contrast between the two techniques, indicating that the higher resolution information brings a higher resolution to the relative abundance mappings. So in key conclusions, the ORF prediction for LoopSeq+CappableSeq methods, it outperforms the ORF prediction in the uncorrected PacBio and nanopore reads. recall that the results did show with correction, which is a fairly computationally intensive process, you can normalize that to some extent, but it never quite reached the LoopSeq performance. We reduced the erroneous mappings of transcripts to closely related strains, again, with more information captured per each one, and that the relative abundance of the transcription mixtures generally improved through the LoopSeq plus CappableSeq methods. And so just acknowledging the contributors, both colleagues at Loop, as well as NEB, Lawrence and Bo from NEB, and then the Kyle and Tuval at Loop, as well as a number of additional colleagues that are shown below. So happy to take any questions. Again, reiterating that this was the same technology, just extended to a different application to take advantage of the LoopSeq method to gain information per transcript and therefore gain accurate mappings and therefore accurate quantitations across the board.